We're going to be discussing a number of important and recurring topics today. We're going to be discussing transmembrane proteins and the concept of diffusion. As part of this, we're going to be discussing a few different categories of transmembrane proteins: channels, receptors, and pumps, all of which will be coming up again in future lessons. And the better we learn them and the more repetition we have, the better you'll understand the concept when it really counts. Of course, if we're discussing transmembrane proteins, you got to start with a membrane. So we're going to be drawing a plasma membrane. Of course, these are the polar phosphate heads and the nonpolar fatty acid tails. And we're ready to draw our first protein. I'm going to be drawing these in green for this particular lesson. And I'm just going to draw a simple one at first, just a generic transmembrane protein, giving you the idea that there's a protein that can make its way all the way from the outside of the cell, which again is considered above, all the way to the inside of the cell, which is considered below. When we draw a phospholipid membrane side to side in a horizontal fashion, the outside of the cell, extracellular fluid is going to be up above. The intracellular fluid, or the inside of the cell, is below. So transmembrane protein goes in to out, or out to in, depending on how you look at it. So a few more phospholipids there, that phospholipid membrane. Now we're going to draw our next protein. This is going to be a channel. A channel is going to have, in this case, in our simplified model, it's going to have 4 component parts. The one in the middle there is kind of sticking out at you, and there's the back of it kind of just peeking there. And then the 2 sides, of course, buried in the plasma membrane. This is a channel. It's basically a straw or a tunnel that leads from one side to the other and allows structures through, either into the cell or out of the cell, that wouldn't normally be able to pass through that bilipid, nonpolar, hydrophobic atmosphere. So we're going to use sodium ions, which I'm coloring in pink, and putting a little plus sign in there. Na+, sodium ions, always exist in higher concentration outside of the cell. And you'll notice they're in very low concentration inside of the cell, so the natural propensity is for the sodium ions to move into the cell, if at all possible. And if they are able to find a channel, such as the one we're looking at here, which is apparently permeable to sodium ions, then they're able to do so. We've just covered a super important concept, the concept of diffusion. Now, the formal definition of the diffusion goes something like it's the net movement of particles from an area where they are highly concentrated to an area where they are less concentrated. But for the sake of simplicity and the importance of this concept of really embedding it in your learning, in your memory, you need to simply describe it as high to low. Literally 6 letters, high to low. Things move from an area where they are highly concentrated to an area where they are less concentrated. Simple diffusion takes no energy. Things naturally do this. And again, normally a sodium ion, which is charged and therefore basically polar, would not be able to pass through an area which is nonpolar, which the plasma membrane is. But if they find a channel, yes, they would be able to pass through that channel. Now, this is a particular type of diffusion called facilitated diffusion. The diffusion of particles from high to low concentration through a channel is facilitated diffusion. What we're going to do now is we're going to take a little slice of this and take a closer look at all of these structures. And therefore, we start with our plasma membrane in order to orient ourselves to this. So we're going to draw a big old phosphate head and some big old fatty acid tails. And now I'm going to draw a protein in closer up view. If you go back a couple of videos ago, I was talking about the hierarchy of protein folding. So what I'm drawing here, I know it looks awful, but I'm drawing some alpha helices in order to indicate this is, of course, the secondary folding of the amino acid structures. And then I'm drawing some larger size sodium ions so that you can sort of see them moving through. Basically, it's like a tunnel or like a straw. Since the sodium ions are charged, positively charged, that's what the plus means, That means that any amino acids adjacent to them, or the bulk of the amino acids that are adjacent to them, must be hydrophilic or polar. Whereas the ones I'm labeling here, the ones that are lying against the fatty acids, must be hydrophobic, nonpolar. They're fatty. They only like being around each other. Fatty acids only like being around other nonpolar things, other hydrophobic things. Therefore, the amino acids in this portion of the protein must also be hydrophobic. Whereas, again, the amino acids that make up the bulk of that alpha helix that is adjacent to the sodium ion tunnel, they are going to be hydrophilic or polar. I know some of you are going to get annoyed because the writing is so small, but we have covered these terms before, so I just wanted a little reminder there. Hydrophobic and hydrophilic, and here's another set of another portion of the protein that I'm drawing. And of course, since this is also adjacent to the tunnel, this would also be, these amino acids the bulk of them would be hydrophilic or polar. Again, if it's next to a charged structure, it must be hydrophilic, it must be polar, or the sodium ions would never be able to pass through that region. And again, the structures lying against the fatty acid tails must be hydrophobic or nonpolar. You think it's fun to draw phospholipids? I think it's kind of fun. It gets a little tedious after a while, but it looks neat at the end, doesn't it? It really looks like the way it's supposed to look. All right. The next protein we're going to be drawing, don't start drawing it too fast. You got to put a little divot in the top there. This is a typical way that we draw this particular structure. This, my friends, is a receptor. Now the receptor, oh my gosh, so important for many physiological responses in your body. Receptor, it's actually right in the name there. A receptor receives something. So that little divot there is called a binding site. The structure that's attaching to it is some sort of free-floating thing, perhaps a protein, perhaps a hormone, perhaps a neurotransmitter, and it fits perfectly inside of that binding site. And it's so important, and oh my gosh, it's going to come up over and over again. You really have to understand it. This receptor is a protein. All receptors that we discuss in our class are proteins. They might be glycoproteins, which means that they have carbohydrates attached, but we usually don't draw them in that level of, we don't discuss them in that level of detail. We usually just describe them as proteins. Now, this particular receptor is going to cause something to happen inside of the cell. The advantage that we have with receptors is that structures, such as polar structures that might have a hard time entering the cell, can stay outside of the cell. And by attaching to this receptor and sending a signal through the receptor into the cell, it's able to change myriad physiological functions inside of the cell, oftentimes through G protein coupling, adenylate cyclase, cyclic AMP, and then the magnification of signal. We are going to talk about this down the line. But oftentimes it actually doesn't take that much signal on the outside of the cell to cause like thousands of events inside of the cell, which is why sometimes hormones, which bind to receptors oftentimes outside of cells, just as what you're seeing here, why hormones can oftentimes have many different physiological effects on the body. The reason for it is that the receptors are sensitive. They magnify a small signal into big changes inside of the cell. So now what we're going to do is take a chunk of this receptor. The most important part would be the part of the receptor that actually has that binding site. So we're going to draw just the tippy top of the phospholipids that are embedded around this protein. There's our phosphate heads, fatty acid tails, just the very top. And now I'm going to go ahead and draw, rather than just a green line, I'm going to go ahead and draw the amino acids. And this time I decided to take a shot at drawing a beta sheet. Remember, alpha helices or beta sheets, these are the secondary foldings of the proteins. And now, I asked my husband about this. I drew a kind of a preliminary draft of this, and he said it kind of made sense. So I'm going to go ahead and go with it. But what I'm drawing up there at the top is basically a receptor, the binding site of the receptor that's shaped a particular way. And it's hard to get across how messy this process can actually be. So I drew a number of beta sheets, alpha helices, and so on. But the most important thing we're drawing right now is the binding site, because it's going to fit the next structure that I'm going to be drawing. So what we have here, believe it or not, it's just awful. But look, we have to make a point here. This is that little brown circle that I drew up above. I'm blowing it up so that you can see that it has a particular shape to it. And the shape of this, there's always little bits that stick out from whatever structure you're looking at. Perhaps it's a hormone, perhaps it's an antibody, perhaps it's a virus, just depending on what the situation is. You'll have binding sites that receive it, and then the free-floating thing will have a particular shape that will perfectly complement, that will perfectly fit into the binding sites of the protein that we're drawing. So yeah, I know it's hideous. Some of us are blessed with artistry. I am not one of them, but I have to get my point across. You must draw with me. You must draw with me. This is how you will learn. Active learning. It's the way to go. Even if you're learning from an absolute neophyte, awful artist. More phospholipids. Phosphate heads sticking into the cell and out of the cell where there's a polar water-filled fluid, both in and out. And then, of course, the fatty acid tails that are most comfortable snuggling in there with each other. Can't tolerate the water. Let's move on to our next protein. Okay, we're going to combine the two things that we just looked at. You'll notice that there is a binding site on the outside of this. Right in your mind, right away, it should be triggering the idea that this is a receptor. That binding site always means receptor of some sort. And then we have 4 transmembrane regions here, which indicates that it's probably a channel. So sometimes it is the case that you'll have a receptor actually directly attached to a channel, which means that this is the simplest way that receptors can work. In this case, a receptor binds to the binding site, the channel opens. So a channel that's normally in a closed state, for example, not allowing sodium ions through, for example. Once the neurotransmitter, say acetylcholine or acetylcholine binds, sodium ions begin to influx into the cell. So we will be coming across those again in future. More phospholipids, and we move on to our next protein. We're going to be, so the outline is sort of angled in this case. It's kind of angled backward. What the heck? Is that a divot? Is it a binding site? Yes, it is a binding site, but this time you'll notice it is on the inside of the cell. And look at this, a number of other binding sites. And we're going to flap another portion of the protein over, angled in the opposite direction. And these are more binding sites, but you'll see that at the moment they're closed. And I'm going to go ahead and label this as a pump. The pump that we usually discuss when we're discussing hypothetical pumps is the sodium-potassium pump, and therefore that is the one that I have drawn here. A pump uses active transport. A channel is passive. It's called passive diffusion. It's called facilitated diffusion. But a pump is the opposite thing. A pump requires energy and allows things to happen. So we'll show that process now. As I had done earlier, we're going to draw a number of positively charged sodium ions outside of the cell. This is, in fact, how cells exist physiologically. A small number of sodium ions inside of the cell and a large number of sodium ions outside of the cell. So what's going to happen is a pump is going to take sodium ions from inside of the cell, where they are less concentrated, and pump them out. Now this pump is going to have to get energy from somewhere, and it's going to get activity, in this case, from a structure called ATP, adenosine triphosphate. We will be discussing this in the future. For now, I just kind of want to draw it so that you understand. ATP is going to bind to this internal binding site. There it goes, binds into the binding site. It fits perfectly. It has a shape that fits and complements that binding site perfectly. Look at this. I forgot to draw the fatty acid tails. I'll come back to those later. The obsessive students will be saying, where are the fatty acid tails? They're coming. They're coming later. I just forgot them. All right, so now what we're going to do is do the opposite thing that we did before. We're going to take this same sodium-potassium pump, and we're going to invert it. And look, here's the binding site. I had to kind of add it in there. Don't forget the binding site. Okay, so the first time we drew it, we drew it sort of like an upside down A. Like an A without the line across. And now we're going to draw it like a V. We're drawing the same thing, but a different configuration. This has experienced a change in configuration. What has caused the change in configuration is the binding of ATP, and then where you have inorganic phosphate that has been left behind, dissociating from the ADP. We're going to talk about that down the line. As I said, what I want you to understand for now is that the action, the change configuration that you're seeing here is generated by the hydrolysis of ATP. So we've discussed the term hydrolysis before, so you know it's the splitting apart of a structure, and we'll discuss it in a little more detail, but for now understand that this pump is powered by ATP. All right, so now what we're doing is we're drawing the idea that those sodium ions, positively charged sodium ions, that were brought from the inside of the cell. Remember, they were smaller in number inside of the cell. They were smaller in number inside of the cell. They're now being pumped to an area where they're more highly concentrated. This is the opposite of diffusion. A pump takes things and drives them in a direction where they are less comfortable. So the change configuration you're seeing here is where in its low energy configuration, it was facing the inside of the cell. Now it's going to be facing the outside of the cell. And acute observers will understand that now those two binding sites that previously were closed are now open. This is one of the benefits of drawing things for yourself. These are the sorts of figures while you're looking at a textbook or Googling the figure, for example. These are the little details that you would miss over unless you kind of forced yourself to draw the thing yourself. So what are those newly open binding sites for? Well, they are for these other circles with pluses in them, positively charged potassium ions. Now, potassium ions, we haven't discussed them before, but they actually exist in higher concentration inside of the cell and in lower concentration outside of the cell. Quick quiz question. If there was a channel that was permeable to potassium ions, would the net movement of potassium be inward or outward? The answer is outward. So the first picture we're drawing here, the A without the bar across the center, is the inward facing configuration. And what happens here is the potassium is going to leave out of its binding sites in the sodium potassium pump and get pushed into the cell. Now this is against its concentration gradient. Remember that potassium is lower concentration outside of the cell, so it's difficult, if you will, if you want to personify this, it's difficult for potassium to move into the cell. Just as it's difficult for sodium to move out of the cell because it's in higher concentration outside of the cell. There's the fatty acid tails. There they are. As we draw the last of our phospholipid membrane, it's worth taking a quick review of what's happening here. The first protein that we drew was some kind of a hypothetical nonsense transmembrane protein. I'm sure it has some purpose, but we haven't said what. Next, we drew a channel through which, in this case, sodium ions were able to enter the cell because they were flowing from their high concentration to their low concentration using diffusion, and this in specific would be considered facilitated diffusion because it uses a channel. The third protein that we drew was a receptor where you had some sort of neurotransmitter, hormone, some structure coming along and fitting perfectly into that binding site to allow for some sort of activity to occur inside of the cell, for example, some kind of a cellular change to occur. Oftentimes, hormones and neurotransmitters are too large to fit through a plasma membrane or too large to fit through a channel, so they simply cannot enter the cell. So this gives them the ability to effect changes inside of the cell without actually having to enter the cell. That's how external receptors work. And the next protein we drew was a receptor channel, basically putting those two concepts together, where you have the direct binding of a neurotransmitter, for example, to a binding site, which allowed the channel, which had been closed, to pop open, change its configuration, and allow whatever thing through, perhaps sodium ions, to flood into the cell. And the last protein we're drawing in two different configurations, inward facing and outward facing. I'm now going to add the words low energy configuration, and that represents the inward facing guy, and high energy configuration. You're looking at the same protein, the same pump. The difference being that when it's in its high energy configuration, when ATP has been hydrolyzed and has allowed for the shape change that allows that V, where it's facing outward now, well, the basic purpose is right in its name. It pumps things against their concentration gradient. And I'm writing it here. It goes against the diffusion gradient, the concentration gradient. Rather than going down the diffusion gradient, it's going up the diffusion gradient. It's kind of going backwards. It's taking structures from an area where they are less concentrated and pushing them into an area where they're more highly concentrated, where it's not as natural a fit. The net movement should be from high to low, which is the standard definition of diffusion. But pumps do the opposite thing. They take things from a low concentration and push them into an area where they are more highly concentrated.